Dear students, happy to meet you on this online mode of education. I am Dr.S.ANGELINA, GLORITA PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M. College for Women, NAGAPATTINam. The topic I am going to discuss with you today is about the pearl oyster farming, which is coming under the paper Economic Zoology. Pearls are closely linked to human life since prehistoric time. Evidences suggest that pearl shells were used as decorative objects during ancient times in Egypt. Pearls are also featured in religious texts like Bible and Quran. Also, the origin of pearls is explained in mythological and cosmological terms. During the reign of Alexander the Great, there was a marked increase in the trade and transport of pearls to the Western world from the Red Sea and the Middle East. Pearls were symbolic of power and purity during the Middle Ages, and many royals adorned themselves with pearl jewels. When we see the history of the pearl, an important period in pearl history were the first voyages of Christopher Columbus and other Spaniards in the 15th and 16th centuries, when they discovered pearls and pearl oyster banks in Central and South America. This generated great wealth for the Spanish crown and triggered what is known as the Pearl Age among us European royals and aristocrats. Although there were numerous attempts at culturing pearls, the first to successfully achieve this feat and market a product was Mikimoto using the Mais Nichikawa method in 1916. With the help of Japanese expertise, cultured pearl farming expanded to other countries. The chief sources of pearls are marine pearl oysters. However, the freshwater mollusks are also known to produce pearls, but they are of poor quality and almost worthless. A pearl, due to its delicate appearance and shine, has retained its position as a costly ornamental object. Due to its properties, it occupied important place in the crown of great emperors and queens. Pearls have been known to mankind since the beginning of civilization. They are highly esteemed as gems for their beauty and splendor. These structures are secreted by the mantle, that is the skin of pearl oysters, in response to irritations caused by external or internal stimuli such as sand grains, mollusk eggs, parasites, DTS, and other foreign particles. Many attempts have been made to culture pearls in freshwater mussels also. When we see the classification of pearl oyster, especially Pinctata fucata, it is coming under the phylum Mollusca, class Bivalvia, order Pteridia, family Pteridae, genus Pinctata, species fucata. A pearl is a hard object produced within the soft tissue of a living shelled mollusk. Just like the shell of a mollusk, a pearl is made up of calcium carbonate, which has been deposited in concentric layers. The ideal pearl is perfectly round and smooth, but many other shapes of pearls also occur. The finest quality natural pearls have been highly valued as gemstones and objects of beauty for many centuries, and because of this, the word pearl has become a metaphor for something very rare, fine, admirable and valuable. The most valuable pearls occur spontaneously in the wild, but they are extremely rare. Cultured or formed pearls from pearl oysters make up the majority of those that are currently sold. Pearls from the sea are valued more highly than freshwater pearls. Imitation or fake pearls are also widely sold in inexpensive jewelry, but the quality is usually very poor, and generally speaking, artificial pearls are easily distinguished from genuine pearls. Pearls have been harvested and cultivated primarily for use in jewelry, but in the past they were also stitched into the lavish clothing. Pearls have also been crushed and used in cosmetics, medicines, and in paint formulations. When we see the distribution of pearl oysters beds, the main sites of production of pearl are the Persian Gulf, Gulf of Mannar, Ceylon, then Sulawesi near Philippines. Besides these, coast of Australia and shores of Central America. Nowadays, Japan has surpassed all the countries in the bulk of pearl production by invention of pearl culture techniques. When we see the pearl fisheries in India, pearl oyster beds in India are present on both eastern and western coasts. However, the east coast is more productive and extensive than the west coast. The two zones of Indian coast known for pearl oysters are Gulf of Mannar and Gulf of Kutch. The Indian pearl fisheries in the Gulf of Mannar along with Sri Lanka produces true oriental pearls of the finest quality. The species common to this area is Pinctata fucata. Gulf of Mannar has 72 beds. Gulf of Kutch, in those regions, there are about 42 pearl oyster reefs known as khadas. Let us see how the pearls are produced. Pearl is secreted by the mantle of the pearl oyster as a protection against foreign objects which may be sand particles, minute larval forms or other such things. To study the mode of formation of pearl, it is essential to know the structure of the shell and the mantle. Shell of the pearl oyster is composed of three distinct layers. They are periostracum, prismatic layer and nacreous layer. Let us see about the periostracum. It is the outermost greenish brown thin translucent layer made up of an organic substance, concholine. It is secreted by mantle. It serves to protect the underlying layers from harmful effects of weak carbonic acid in water. The next layer is prismatic layer. It is the middle layer secreted by mantle. This layer is made up of minute crystals of calcium carbonate. It gives strength and rigidity to the cell. And next is about the nacreous layer. It is the innermost layer of the shell and is better known as mother of pearl because this layer is responsible for the formation of pearl. It consists of alternative layers of calcium carbonate and concholine. This layer is also secreted by mantle and its function is to protect the delicate surface from the harmful effects of the foreign particles. That pearls are formed due to irritation caused to the mantle by the entry of foreign bodies. Whenever any sort of foreign body enters and gets between the mantle and the shell, it becomes enclosed in a sac of mantle epithelium. This foreign body now acts as an irritant and stimulates the mantle epithelium to secrete concentric layers of nacre around this foreign body. These layers then hardens, it becomes pearl. The size of pearl is directly proportional to the degree of irritation caused by the foreign agents. Time taken for the formation of a pearl average size is 3 to 5 years. Actually, in the Gulf of Mannar, the diameter of the nucleus is 3 mm and the diameter of the pearl is 3.63 mm. And also time taken for culture these pearls, it will take around 6 months and 11 days. And also in Japan, here the diameter of the nucleus 3.05 mm and the diameter of the pearl is around 3.70 mm. And also the time taken for the formation of these pearls, it will take around 2 years. The chemical analysis of a pearl shows that it contains about 90% of calcium carbonate, 5% organic substances and 5% water and other residues. The natural pearl is rarely a sphere as it assumes the shape of the nucleus. The process is very slow and continues for the whole life of an oyster. The value of a natural pearl depends on the size, shape, color and luster. Let us see something about the biology of the pearl oyster. The oyster usually breeds several times in succession. The larvae are free swimming and feed upon microscopic organisms. They undergo metamorphosis and finally settle at the bottom in large numbers. The young oyster has a fast growth rate and attains about 50 mm size in a year. Pearl oysters feed on small algae found in the water column through filtering large amount of water. Let us see something about the formation of pearl. If a foreign particle and insert material, example sand particle, hard object, living object, anything like parasite, it penetrates into the mantle of the oyster, the cells of the mantle gets attached to it. The foreign particles forms the nucleus over which more or less concentric layers of mother of pearl is secreted. In case the particle penetrates deep enough, a sac or cyst is first formed around the particle and pearl formation occurs within the sac. With time, due to more secretion, it enlarges in size and the pearl is formed. Artificial pearl culture, when we look into it, Japan was the first to develop scientific pearl culture in 1907. Introduction of large particles of desired shape and size made it possible to get pearls of different shapes and size. Forming of pearl oyster is essentially for meeting two demands, that is, to grow the wild collected hatchery produced spats to implantable sized oyster, to grow the nucleated oysters for pearl production. When we see the life cycle here, the eggs and sperm getting fertilized and it releases the fertilized egg, that hatching, it will take 15 hours, then it releases the free swimming trophophore larva, then this trophophore larva, it converts into veliger larva, this is also free swimming, then it converts into pediveliger larva within 20 days, then the metamorphosis continuously taking place and it transforms into juvenile pearl oyster that develops into adult pearl oyster. When we see the oyster sources, the basic stock for the forming operations is either collected from the natural beds or from the hatchery. There are about 65 pearl beds have been identified in the Gulf of Mannar, area of Tuticorin at a distance of 12 to 20 km at the depths ranging from 15 to 20 meter. The availability of the oysters in these beds is erratic due to the irregular spat settlement. Pearl oyster spats can also be collected to smaller extent by suspending various kinds of spat collectors for forming purposes. However, such natural collections may be of different species composition which sometimes may not be useful in production of pearls. Hence, an established hatchery should be a sustained source for spats for culture operations on continuous basis. When we see the site selection criteria, the selection of farm site is very important. The selection would be based on the biology of the organism formed and the ambience of the prevailing environmental parameters of the site. Congenial conditions such as protection, water current, clarity, optimum temperature and salinity regimens to be considered while selecting the farm site, apart from the site being free from any kind of pollution. A deep sheltered bay, protected water bodies within the sea conditions, not going too rough, offers an excellent site for pearl oyster forming. Technological and economic considerations play a major role in the selection process. A careful appraisal of the habits of the organism to be cultured would give a reasonable level of confidence on the tolerance limits within which the various environmental parameters can vary. Due to the consideration has to be taken given to possible effects of fluctuating water flow, primary production, siltation, etc. in order to obtain the optimum level of growth and production of high quality pearls. Unsuitable levels of environmental factors such as salinity, water temperature, cold water currents and other factors such as red tides, hydrogen sulfide and pollution by industrial and domestic effluents are serious hazards to pearl culture. Sheltered bays are ideal locations for pearl oyster farms. They offer good protection to the culture structures such as wraps and cages. Shallow coastal waters where the sea is calm most of the year can also be considered as a suitable site. When we see the environmental conditions, let us see some environmental factors. Number one is temperature. In temperature regions, The water temperature plays an important role in the biological activities of pearl oysters. In Japan, the optimum temperature for oyster growth has been found to be between 20 to 25 degrees centigrade. A temperature below 13 degrees Celsius causes hibernation. Below 6 degrees centigrade, the oysters die. At temperatures above 28 degrees centigrade, the oysters show exhaustion. The thickness of the pearl oysters are affected by the minute changes in water temperature during the day and also vary considerably according to the season of the year. The deposition of calcium stops at the water temperature of 13 degrees centigrade. In the Gulf of Kutch, the oysters grow vigorously in winter months when the seawater temperature ranges between 23 to 27 degrees centigrade. A slight decrease in temperature triggers spawning in oysters in the Gulf of Mannar. The growth temperature relationship is valid only up to a certain temperature for optimum growth. Next is about the salinity. Pearl oysters tolerate a wide range of salinity from 24 to 50 units for a short duration of 2 to 3 days. Salinities of 14 and 15 units may cause a 100% mortality among the oysters. The effect of salinity on the growth of the pearl oyster has not been clearly investigated. However, it appears the pearl oysters tend to prefer high salinities. Oysters raised in such salinities produce pearls with a golden tint. Bottom is another factor. Gravelly bottom are suitable for pearl oyster forming, while sandy or muddy bottom should be avoided. Oyster growth is affected by water temperature and nutritional condition of the ground. Repeated culture on the same ground leads to some extent the deterioration of pearl quality. The chemical and physical state of the sea bottom is affected by the organic substances discharged from the oysters and fouling organisms. Periodic removal of such accumulated substances from the bottom of the culture grounds often increase production as well as quality. The next is about the depth. The optimum depth for forming pearl oysters is around 15 meters. At greater depth, even if the rate of nacre deposition is slower, pearls of high quality with a pinkish coloration are obtained. In slick load, pearl oysters generally prefer clear waters as high turbidity levels will affect their filtration efficiency. A decline in oyster condition was noted at Repaladi Farm due to the high silt content in the farm area throughout most of the year. Next is water current. The current sites, the culture sites should be naturally sheltered against strong winds and waves. Tidal amplitude and currents must be sufficient in order to allow oxygenated water and fresh plankton and flush away waste materials. In strong water currents, the formation of the pearl oyster is usually fast, but the quality of pearls produced is affected. The next is primary productivity. The condition of a specific culture ground depends primarily on the chemical constitution of the seawater and on the species and amount of plankton present. Rich nutrients discharged by a river into the sea are responsible for high primary productivity. The oysters probably derive their chief source of concholine from the nitrogen substance of the plankton. The organic matter and calcium dissolved in the seawater are directly absorbed by the food consumption cells. The calcium passes through the mantle to be deposited on the surface of the shell or pearl in the process of their formation. The presence of trace metals in small quantities influences the color of the nacre. When we see the sources, the basic stock for the forming operations is either collected from the natural beds or from the hatchery. There are about 65 pearl beds have been identified in the Gulf of Mannar area, Tuticorin at a distance of 12 to 20 kilometers at depth of ranging from 15 to 20 meters. The availability of the oysters in these beds is erratic due to the irregular spat settlement. Pearl oyster spats can also be collected to similar extent by suspending various kinds of spat collectors for forming purposes. When we do the supply of oysters in pearl oyster forming, oysters collected from the natural beds or reared from naturally collected culture spat are used. In the Gulf of Mannar, several pearl banks are distributed in the Tuticorin region at the distance of 12 to 15 km at the depth of 12 to 25 meter. Pearl oysters from these beds are collected by skin and scuba diving. Wide fluctuations in terms of pearl oysters availability have been noted in different pearl banks in this area during the last few centuries or also during the most recent years. In the Gulf of Kutch, the pearl oysters are found on the intertidal flats and the population in sparse. Collection is done by hand. In Japan, oysters fat are collected by submerging bundles of cedar twigs near the water surface during the peak larval settlement season. Hides, flims and old fish nets are also commonly used as fat collectors. Almost the entire requirement of oyster supply to the pearl culture industry is met by this type of spat collection. Spat collection attempts in India have not been successful, and this may be due to the distance of the pearl oyster beds from coastal waters. However, India has recently succeeded in producing pearl oyster seed under hatchery conditions, thereby providing the industry with a more dependable source of oysters. Let us see something about the methods, rearing methods. Raft culture. First, the raft culture is considered to be one of the most suitable farming methods in sheltered bays. The size of the rafts can be altered according to the convenience of the situation. A raft of 6 into 5 m in size can be easily constructed and floated with 4 buoys. Rafted are usually constructed with logs of the teak, venteak or casuarina wood or chosen length with a bottom of about 10 cm diameter, tapering to 6 cm diameter at the tip. These logs are arranged as per the requirement and lashed with quiet ropes. Floats are attached to the raft to give buoyancy. The floats can be sealed empty diesel drums of 200-liter capacity with fiberglass coating, mild steel barrels painted with anti-saline and anti-corrosive paints. Unit raft system is found to be convenient and well suited to the Indian sea conditions. Rafts are moved with anchors at opposite sides with tested quality chains, and their direction is decided according to the prevalent wind direction at a specific site. This is a more suitable method to form pearl oysters in the sheltered bays with considerable depth. Rafts are constructed with teak wood poles of 4 to 5 inches thickness or sliced timber of similar thickness. The teak wood poles are lashed with coir ropes, and the sliced timber poles can be fixed with a steel bolt and nut. Rafts are generally almost square in shape. Such constructed rafts are floated with the help of 4 buoys and moored by 2 anchors of 30 to 40 kg weight. Appropriate anchor chains are provided after considering the depth of the area. Empty oil drums coated with fiberglass, mild steel barrels or few alternates to serve as floats for a raft. Next is about the long line culture system. Here, it is practiced in the open sea where the depth is more. This is more suitable to withstand the high wind and wave action. Long line is primarily constructed with a long synthetic rope 15 to 20 mm with the 2 main floats on both ends. Depending on the length of the long line, smaller floats is also attached to them. In the long line culture method, spherical or cylindrical floats which are connected by horizontal synthetic rope or chain are used. The oyster cages are suspended from the ropes. This system is good for open sea conditions. In another method of hanging, a hole is drilled near the hinge of the pearl oyster. A small thread is put in through the hole, which is then tied to a straw rope coated with tar. The straw ropes are hung from a raft. The next is about the on-bottom culture. Here, sea bottoms with the granite or coral stones composition can be used for on-bottom culture. In the Tuticorin Harbour Basin, where the brackish water has been constructed with granite stones, the protected portion of the breakwater is used for culturing mother pearls. 1 m of water is available below the low water mark. Then, due to constant circulation of seawater, settlement of fouling organisms is poor and inconsistent. However, it has been noted that the growth of the mother oyster is lower in on-bottom culture compared to the growth of cultures cultured in raft. The next is about rearing containers. Culture of mother pearls. Here we can use the box cages measuring of 40 into 40 into 15 centimeter are used to rear mother pearl oysters. The size of the mesh varies with the size of the oyster to be reared. The frames of the cages are made up to 6 mm mild steel rods coated with anti-corrosive paints or coal tar. Box cages are useful in general mother oyster culture. To trace the history and performance of individual oysters, frame nets are used. The frames measuring from 60 into 40 centimeter, each with 5 compartments, meshed and hinged at one end, open as a book. The oysters are arranged in rows and held in the compartments when closed. The space available in between the two frames is about 10 mm, which is sufficient for the oysters to open their walls for feeding respiration. And in the juvenile rearing, the pearl oysters are reared in net cages, synthetic fabric of Villan screen bags, whose sides are stretched with a steel rod in the form of a prism, also used for rearing of juveniles. The mesh size of the screen depends on the size of the juveniles to be reared. The mouth of the bag is tied with a synthetic twine, which facilitates opening or closing when required. To provide further protection and the predators, the bags are placed in old nylon net bags. Clogging by slit and the growth of the fouling organisms can be prevented by periodical replacement of the Villan screen bag, which it can be cleaned, sun drained and reused. Spats of up to 2 centimeter in size are reared in these small net cages. Box cages, which are used for rearing mother oysters, can also be used for juvenile rearing by providing an additional Villan screen cover inside the cage. Farming system in India, the farming practices of pearl oyster is very much limited to the two Gulf zones, that is Gulf of Kutch in Gujarat and Gulf of Mannar in Tamil Nadu. The method of farming varies to the prevailing farm site conditions. Accordingly, the following methods are developed and practiced. When we see the farming of pearl oyster is essentially for meeting two demands. To grow the wild collected, hatchery produces fats to impartable size the oyster. To grow the nucleated oysters for pearl production, then oysters have 3-chambered hearts that pump colorless blood throughout their bodies. They breathe with gills just like fish. Wild oysters can live 25 to 30 years. Natural collections may be of the different species composition, which sometimes may not be useful in production of pearls. Hence, an established hatchery could be the sustained source for spats for culture operations on continuous basis. Onshore culture system, here the system of recent origin. Seeds of pearl oysters are reared in cement tanks of size ranging from 250 to 500 square meter. A water level of 1 meter is maintained. Oysters are held above the bottom through a grid system constructed using PVC pipes. A stocking density of 125 numbers per square meter is maintained. Raw seawater is pumped into the settlement tank and then to the rearing tanks. Stocked oysters are supplemented with mixed culture of many species. Cell concentrations in the rearing medium is maintained at 75,000 cells per liter in a tank. Daily 25% water exchange is required. By this method, an average growth of 50 mm in 6 to 7 months from the stocking size of 5 mm is achieved. Culture containers, fat transplantation cages. Here, this type of cages is fabricated using the box cage iron frames. Suitable sized Villan screen bags are prepared and the cage frame is inserted inside both the ends of the region. Old fish net is stitched as bag and rolled over the Villan bag. This provides an extra strength to the spat cages. A central rope is provided and flooded in the center so that the cages are hang horizontally in the culture structures. A stocking density of about 1,000 numbers per cage can be maintained for a short period. The small meshed Villan screen bag has to be replaced with large meshed Villan screen bags every 15 days at the spat growth. Clogging takes place very quickly in these cages and hence a periodical cleaning is very essential to avoid mortality. And in box cages, oysters of above 30 mm are reared in box cages. Box cages are made of 6 mm steel rods, square in shape and measures, and frames are covered with suitable sized old fish nets and stitch. Suspense ropes are provided for tying to the culture structures. 150 to 200 numbers of oysters with 30 mm can be reared in such cages. Then book type cages, here they are made of 2 rectangular frames stitched with the required size netting. Many subsections are provided to hold oysters in series, meshed and hinged at one end, open as a book. The arranged oysters are held in the compartments when the cages are closed. The space between the frames is 10 mm and sufficient oyster are placed in that cages. These cages are designed for holding oysters for experimentation. Then latent cages, here they are fabricated using circular iron frames of 4 to 6 mm rod arranged in 2 to 3 layers. Plastic sheath can be provided over the iron rod for longer life of the cages. Appropriate sized mesh is stitched over the frame and opening is given at each layer to handle the stocked oysters. The cages are vertically hanged in the culture structures. Then in the farm management, major problems in the maintenance of pearl oyster stocks in the form are the biofouling organisms. These organisms settle and grow on the shells. The boring organisms riddle through the shells and render them weak and fragile to predators. These factors, either singly or combinedly, can affect the formed stocks or result in growth retardation and mortality. Fouling, the most dominant fouling organisms in pearl oyster farming is barnacles. Here we can see the settlement on the surface of the pearl oyster, the profuse growth of sponges, Calispongia and many other sponges, they will attach on the oyster cells. The next is boring. The dominant groups of boring organisms are the sponges and polychaetes. And here we can see the result. The furrow becomes deeper by the accumulation of mud and thus causing a peeling of peristomium, making the shell weak. Such affected oysters suffer great physiological strain in respiring the shell and later leading to mortality. Boring mollusks such as Lithophaga species and Mortia species, which make holes on the shells, are also occasionally found on the oysters. Next, something about the predators. The gastropods murex, which is an important predator of the formed oyster. They can consume at the rate of 1 oyster per day per animal. Crab species also prey upon oysters in the form. Sometimes the Xanthus species are also found to prey the oysters in the cages. The boring and fouling organisms can be killed by applying a number of chemicals in appropriate dosages on the individual oysters. However, in the farming conditions, the best method of controlling the boring and fouling organisms on the formed oyster is thorough periodic cleaning by scraping of the borers and foulers. Predatory mollusks in the cages can be picked and killed. By following the judicious choices of depth for growing oysters can also avoid such problems as fouling in deeper waters relatively less. Natural enemies of pearl oysters are octopus, barnacles, eel. In addition, low salinity of the water caused by the heavy rains and red tide are also dangerous to the pearl oysters. Sometimes growth of the seaweeds and barnacles kill many oysters. When we see the benefit significance, here the pearl farming is an attractive business venture because of the high value of the final product. Although black pearls vary greatly in value depending on their size and quality, large round pearls of high quality can sell for very high prices. In recent years, pearl prices have fallen significantly, especially for smaller, lower quality pearls. The price of large and high quality pearls, however, have not declined as rapidly. One of the principal advantages of pearl production is that the final product is lightweight and non-perishable. Pearl industry in India has a great prospect as compared with the other pearl producing countries that have occupied the world's pearl market. In India, the artificial pearl production has started at the Tuticorin Research Center of CMFRI with the collaboration scheme of the Tamil Nadu Fisheries Department. In 1975, the Alagi Swami has proved the growth of rates of pearls is much faster in India than that of Japanese sea. Pearl oysters are most commonly found in remote tropical atolls where commercial exploitations of marine resources such as fish is difficult due to lack of refrigeration and shipping facilities. Pearls require no refrigeration and very simple processing. Pearl farming is also a compatible occupation for people who like working on the water and have boating, diving and fishing skills. With the exception of the grafting process, pearl farming is a relatively simple form of aquaculture because pearl oysters do not require artificial feeds, complicated farm structures or constant attention. If properly managed, pearl farming will not harm the environment and can increase the wild pearl oyster population over a period of years. For these reasons, pearl culture may be the best opportunity for business development in many isolated island areas. Let us conclude. Successful pearl farming requires a long-term investment of time, money, and hard work. Producing top quality pearls is achieved by taking good care of the pearl oysters during all stages of farming and is also dependent on the skill of the grafting technician. Nowadays, pearl farming in India is gaining momentum, and many farmers have minted huge profits by doing this form of aquaculture business in the last few years. It can be managed with limited human resources and is one of the thriving farming businesses and yield outstanding profits with low investment and less labor. Pearl industry in India has a great prospect as compared with other pearl producing countries in the world. Thank you.